Hi, everyone. Today we are going to be talking about biotechnology. So, now that we know about DNA and how it works, we are going to learn about some applications and how we can use DNA to do different things. So, some things that we might be able to do with DNA include using it for forensics to identify a suspect or to identify a victim. We can use it to make things like insulin or certain foods. We can use it to help us learn more about ourselves, and we can even use it to make some changes to a genome like you see there with those pictures. So, biotechnology. The human genome has a lot of different bases, and reading those bases and understanding how to decode those bases is key to helping us use them to do some of the things we just talked about. So we know those bases are found on chromosomes, like you saw there. So one of the main concepts we're going to be talking about is the idea of genetic engineering, which is when we manipulate DNA. And we need a set of tools to work with if we're going to be manipulating DNA. So, this unit is going to survey some of those tools and some of those techniques we need to manipulate DNA. So, our toolkit, as it says there, it's kind of hidden by that bar. Our toolkit is going to contain some information about bacteria. We already know a little bit about bacteria. Bacteria are one-celled prokaryotes. They reproduce by mitosis, so when they reproduce, it is asexual. It's called binary fission. So binary fission is a simpler process than mitosis. Gives us a similar result, but it's much simpler, right? It doesn't have all those steps, as we know. Bacteria have very rapid growth, so it's easy to work with bacteria since they can make a new generation even in minutes. So we can get a very large colony of bacteria very quickly. Bacteria are the dominant life form on Earth, and they're very diverse, which also is going to help us out so that we can pick what we need to pick in order to work with. The DNA in a bacteria is a single circular chromosome. You see that here. This is that one chromosome. It's haploid because there's one, right? There's not a pair, there's only one, and it's called naked DNA. And what that means is that there's no histone protein. So there's no winding and tightening. There's no making a chromosome versus chromatin. There's none of that. It's just there, free floating. Remember, bacteria don't have a nucleus. In addition to that chromosome, which is pretty long, 4 million base pairs, it's got maybe a couple thousand genes. In addition to that chromosome, we have, um, in addition to that chromosome, we have things called plasmids, which we'll get to in a moment. So we already went over this experiment where we mix the heat-killed pathogenic and non-pathogenic bacteria. And that's because bacteria tend to pick up naked foreign DNA. So whenever bacteria sees some DNA that just happens to be laying around, they might pick it up and incorporate it into their own genetic set. We call that transformation. Bacteria even have surface transport proteins specialized for taking up that DNA. So basically, what the bacteria is doing is it's importing bits of DNA from other bacteria into itself. And it's going to incorporate it into its own chromosome, into its own genome. And then it's going to express any new genes it gets through that process. Again, that's transformation. And this is important because it represents recombination. You might think this seems really risky to be incorporating DNA into your own, but the benefit is that you get some genetic recombination out of it, and you would not get that normally because, as we already know, bacteria reproduce asexually. So in addition to the chromosome, we've got plasmids in the bacteria. These are small supplemental circles of DNA, much shorter than the chromosome. They are self-replicating, and they may carry a few extra genes. So plasmids are separate from the chromosome. We can see them here. They're much shorter. They're self-replicating, and instead of carrying a few thousand genes like the chromosome, they may only carry a few genes, including genes for antibiotic resistance. So if you treat this bacteria with an antibiotic like maybe ampicillin, then it's possible that this bacteria could survive because of a gene it may have on one of its plasmids. Plasmids can be exchanged between bacteria. So this is called conjugation, and this allows bacteria to change pretty quickly and could even lead to evolution. Okay. So how can plasmids help us? Well, plasmids are very simple, they're very easy to deal with, and it's a way of getting a gene into a bacteria easily. We can actually take advantage of this. We can insert a plasmid into a bacteria. We would call that a vector. Then we can force the bacteria to express the gene we just put in that plasmid. And then the bacteria is going to make a new protein. So, for example, what we could do is we could cut a bacterial plasmid, and we can take a gene that we have, might be helpful, maybe a gene to produce insulin, and we can insert that gene, the gene for insulin, into the bacterial plasmid, and then we can stick it in a bacteria, and the bacteria will express that gene. So it's a pretty amazing thing we can do, basically engineering a bacteria to do what we want. So plasmids can be used to insert new genes into bacteria. That's the point. Here's the gene we want. It might be a gene for insulin. It might be a gene for human growth hormone. It might be a gene for lactase. Maybe you're lactose intolerant and could benefit from a gene that will break down lactose. We cut it out. Then we cut the plasmid that we want. And then we insert the gene. You see that right here. We stick this back in the bacteria. We use some ligase to glue it all together. We stick it back in the bacteria, and the bacteria will express that gene and make insulin and make human growth hormone or lactase. All right, so the penguin that you see here is asking an important question. How do we cut DNA? We can't use scissors to cut DNA. It's way too small. Well, we can cut DNA with something really important called restriction enzymes. Restriction enzymes, also sometimes called restriction endonucleases, were discovered a few decades ago, and they're naturally occurring. They already occur in bacteria, and their job is to act like an immune system for the bacteria. They basically restrict the action. Here we have a bacteriophage infecting a bacteria. A restriction enzyme is going to cut this DNA up. So restriction enzymes act as part of the bacteria's immune system. They protect against viruses. They may even protect against other bacteria. And bacteria don't have to worry about their own DNA being attacked by restriction enzymes because they protect their DNA by something called methylation, and they make sure not to have the sequences that restriction enzymes would recognize. Okay, so what do you notice about these phrases? They're all palindromes. They read the same forward as backwards, right? Well, restriction enzymes work in the same way. A restriction enzyme is going to cut in a very specific sequence, and those sequences are palindromes. Take a look over here on the upper right-hand side, GAATTC. And on the bottom, if you read it backwards, it's GAATTC. We refer to these as palindromic sequences, and that is what restriction enzymes look for. That is called a restriction site. And restriction enzymes look for a very specific restriction site, and they cut in a very specific way. You can see that it cuts in between the G and the A. It cuts through the hydrogen bonds separating the two strands, and then it cuts between the A and the G, making two separate pieces. it produces what are called protruding ends, right? The TTAA here and the AATT here will be separated, and they will be sticky because they basically want to hydrogen bond to something, but they've been cut away from it. So it produces these ends that can bind with something else. They will bind to any complementary DNA. So we can put a new piece of DNA in, and as long as it has been cut with the same restriction enzyme and has the same sticky end, we can add in DNA. There are many different restriction enzymes. You don't have to memorize these. EcoR1, HIN3, these are all different restriction enzymes. So there's more than one. Okay, so restriction enzymes, they got these scientists a Nobel Prize. Restriction enzymes, again, to review, cut at specific sites, leaving sticky ends. So here, we're going to cut the DNA into two pieces at the restriction site. Then we can cut the DNA we want with the same restriction enzyme. It has to be the same one, leaving sticky ends on both. Okay, so here's the gene we want. It's on the right-hand side. The end is here, and the green is the gene we are looking for. Here's the chromosome we want to add the gene to. So we're separating the chromosome. And we can paste the gene right in. So the gene gets added to the chromosome. We've basically added a gene that we want to a chromosome, and that's part of genetic engineering. Here's the gene we want, right? What could this gene be? Maybe for insulin, it might be for HGH, it might be for lactase, it might be something else. And we've inserted it into this chromosome. Right? Just like you see here, adding a gene in. Okay, then we need a ligase. Ligase is going to help to join the strands together, just like glue. Okay, gene produces protein in different organism or a different individual. So we could have a bacteria producing insulin, which a bacteria normally is not going to do that. Okay, bacteria making insulin. So how can a bacteria read human DNA? Isn't human DNA different from bacterial DNA? Well, the answer to that question is no. Every single organism pretty much has DNA, and that DNA is the same. It's the same bases, and they decode the DNA in the same way. So even though the DNA of bacteria has a different sequence than the DNA of humans, it's all read in the exact same way. We could read a bacterial gene, a bacteria could read one of our genes because they're all decoded. Every gene is decoded in the exact same way. Okay, so to review, basically, we can insert a recombinant plasmid into a bacteria. We've used restriction enzymes to do this. Then we can grow the new bacteria in a petri dish. The bacteria is going to copy its plasmids, basically cloning the plasmids. The bacteria is also going to reproduce. And basically, we end up with many copies of that gene that we just inserted, and we're going to make a lot of the new protein. In this case, you can see off to the side, it's a form of insulin. So we've basically made the bacteria do what we want by genetically engineering it. Again, cut out the plasmid, cut out the gene, put it in the plasmid, transform the bacteria. The bacteria will grow, and then we can harvest the insulin that the bacteria then make. So there are many uses for this. We can actually use it to transform different things. We can protect crops from insects by making Bt corn. Using a similar process, we can extend the growing season by adding an anti-freezing gene from the flounder into a strawberry. And we can improve the quality of food. In the example here, we've added vitamin A to improve the nutritional value of rice in an area that may have lots of individuals with a vitamin A deficiency. So we can genetically engineer things. In this example, we can actually add a gene for glowing in the dark or glowing under ultraviolet light to pigs. So we take the gene from the jellyfish and we can add it to mice or pigs. We're going to do something similar in a lab. We're going to add a similar gene to bacteria to see if we can make bacteria glow. We'll do that later. Okay. So I'm going to skip that. What are some other uses of biotechnology? So some other uses include DNA sequencing. We can actually use computers and some techniques to figure out the order of bases in an organism's cell. So we can read the entire genome, letter by letter, A, T, T, G, G, C, C. So it can help us discover the function of genes, or it can help us discover why a gene may be malfunctioning. DNA sequencing is pretty important. We can also do DNA fingerprinting. This is when we cut the DNA and we run the DNA through a gel. So we're actually cutting the DNA and putting it in a gel here. The DNA gets pulled through the gel and the gel separates the fragments and we can compare them. Here, we can see we've put different DNA samples into these wells and we turn on this machine and the machine is going to pull the DNA down the gel and the gel is going to separate the DNA by size. So the shorter molecules go further and the longer molecules, pieces of DNA, don't make it quite as far. We're actually going to do a lab about this that shows how this works. So there we see a gel and we see the separated DNA and we can compare it to see if anybody's the same or different. We can also use a special technique called PCR to make more copies of a gene. So down here in the second bullet point, it says, PCR is a special technique. We use a machine to make many copies of a DNA fragment very quickly. It's very fast and it's very efficient. What do you need to do PCR? You need the fragment of DNA you care about. We need free nucleotides because we're going to be making more DNA, so we need many free nucleotides. We need RNA primers, right? We know that in order to replicate DNA, we need those RNA primers already there. So this actually means that we need to know the sequence of the ends of the DNA we want to copy. That's how we get the primers. And we also need something called the Taq polymerase. This is a DNA polymerase that is in a microorganism that lives in very hot environments. To do PCR, you have to heat up the DNA, and basically DNA polymerase is not going to survive that heating process, but Taq polymerase will, because it comes from an organism that is adapted to living in a very hot environment. So basically, you take the DNA and you heat it, and then you cool it, and then you raise the temperature a little bit. So here's our DNA of interest. Here's our primers. Here's the free nucleotides, and here's Taq polymerase. When we heat to a pretty hot temperature, that denatures the DNA. The two strands separate apart. Then when we cool it, that's the annealing part, right? That's step 2. Annealing allows the primers to attach to the ends of the DNA fragment. Then when we warm it up, the Taq polymerase springs into action, attaches to the primer, and starts adding in more nucleotides to extend the DNA. It happens here and it happens here. And now we've gone from one piece of DNA to 2. We do the process again and we go from 2 to 4. And we do the process again and we go from 4 to 8. And we keep going and we keep going, and eventually we end up with more and more copies of DNA. So we use this machine that changes the temperature to make more copies of DNA. And that is all we have for today. We're going to review this and do some labs to help us understand these techniques. So please let me know if you have any questions, and I'll see you next time.